I'm not going to talk what in general the lab is doing, but I'm going to talk about viruses in the permafrost. We've seen that where the permafrost is. We've seen that slide before, and most of the permafrost, it's really amazing. Actually, most of the permafrost and most of Siberia is actually permafrost, and most of it is very ancient permafrost, which gives you access to old times, for which you can actually try to discover something. The bad thing is that actually the permafrost is melting, and is actually melting more in the region of interest, which are basically, we are going to talk about this part of the region right here, which is actually basically warming up 3 times faster than the rest of the planet. So there is some kind of urgency to know what might be in there. There are natural consequences. There are huge holes in the ground, like the very well-known Batagai, which is about 2 hours flight from Yakutsk, which is just the soil is sinking, and this is progressing about 20 meters a year, this huge cliff right here, and nobody exactly know how this started. You have those holes in the ground, which it's still mysterious, but people believe it's some kind of methane explosion, but nobody is there to listen to it, so we don't know where it's actually coming from. And you see those bumps. So there is a lot of very interesting phenomenon that nobody exactly understands what's going on. But even worse, there is now, because of the capacity that we have, because the sea is open of ice for the most of the time now, there is the capacity of building industrial complexes where there is a lot of resources, okay? And there is a lot of resources, right? You know, a lot of oil and minerals that are actually valuable that you can start mining, because now using boats you can actually start up bringing very heavy equipment. And there is those huge mining activities in Russia. This is an old ore. This is in Mirny. This is that was the largest diamond mine. But I did the calculation, and when you exhumate that this is most of that is actually permafrost, okay? So the permafrost that was at the bottom of that hole was probably 1 million year old, okay? You exhumated about 10 to the 20 bacteria. And so apparently nothing happened at that time, or nothing that happened. But there is a lot of things in the soil, and when you make those kind of holes, a lot of things happen. What is even increasing now is because, as you know, the Russians have that capacity now of having portable nuclear power plants, which will actually bring enough energy for about 100,000 people. And then this will make installment of people in those very inhospitable countries very quickly, okay, very, very quick and easy. So this is going to change. So we will heard about the anthrax epidemics now, which is in fact active layer melting, but sometime when the summer is higher than warmer than usual, the active layer is thawing on a larger depth. And then this one was 70-year-old layer that never thawed before, and then because of the very hot summer in 2016, this layer was melting and released anthrax spores. Traces, you've heard about this, traces of smallpox viruses. DNA of smallpox virus has been found in many areas, in many corpses, because in those latitudes, basically, when you put people, when you have people in the ground, they don't decompose, okay, and so they stay basically the way they were when they were buried. And those are 400-year-old, for example, in which you can really very easily find out DNA sequences of smallpox virus, not the virus itself, as it's been solved before. But now, I'm going to talk about, this is where we come in, things which are in fact much farther back in time. Okay, we were not at all interested into this type of subject until I saw that paper in PNAS in 2012, I think, by David Gelishinsky, who is a pioneer of microbiology of the permafrost. that started probably in the 70s, but nobody ever read those papers because they were either in Russian or when they started to publish them in English, they were published in the journal Astrobiology. Because the whole thing was that many of those people thought of the permafrost as a good model for Mars. Okay, and they wanted to show that it was possible that there was life on Mars using permafrost experiments. Okay, so they were able to rescue a lot of bacteria, including very old bacteria. But the most spectacular was the ability to rescue a live plant from permafrost that was 30,000 years old. Okay, 30,000 years old is the end of Neanderthal. Okay, and this is much before the end of the mammoth and the rhinos and a lot of other animals in the Arctic. Okay, so now we have a direct connection, a living connection with very old past. Okay, so when I saw that paper, I said, well, if they can rescue a plant, we should be able to rescue a virus. And so this is what we've done. Am I? Yes. Okay. So now I'm talking about where we work with our Russian collaborators in Turkey. We are in fact based on the very famous Zimov stations, if people knows about it. Okay, this is another place where you are going to spend your winter vacations. Summer is fine. Okay, and this is called Stanislavski Yar, which is an exposure of permafrost that you can actually dig this way, horizontal, which is very good for trying to avoid contamination, because of course if you start digging from the top, okay, you will get all the recent material in your core. Okay, here if you can go sideways, okay, you are going to be able to directly probe the age you are interested in. Okay, so this is what we've done with our collaborators, and at that time we were in fact, because we did discover the first giant viruses that in fact are viruses of Acantamoeba, and Acantamoeba is a very tough protozoan that can actually encyst, so they can also actually survive in permafrost for quite a long time. Okay, what we do is we take a sample, we put that sample in contact with the Acantamoeba cultures, which are been learned to tolerate a lot of antibiotics. Okay, we look for cell lysis. And when we have cell lysis, we isolate the agent and characterize it very thoroughly. Infectious cycle, genomics, transcriptomics, and proteomics. Okay, so we do everything that we can with the virus. And doing this, on one of the samples that was given to us by the Gilisinski lab, we isolated 2 new viruses. Okay, when I'm saying isolating, we have those viruses, they are alive, they are in the lab, we can produce kilograms of it. Okay, so this is not just looking at DNA. Those are very big DNA virus genome. So it's not the part of the talk here that are called giant viruses, but of a single sample, we're able to rescue, to revive 2 new viruses. At the time, those viruses were not known to the world. They were the first member of their new family. Okay, so that was easy to publish, in the sense that we were not accused of contamination because nobody had seen those viruses before. That was lucky, because it is always the case when you try to revive things from the past, as you know about the Neanderthal genome. You have always the accusation, you have always the data that you might have contaminated data by modern samples. So when we did that, of course, it was we catch a lot of media attention. I think we got 300,000 references in Google News. Okay, there was a book that was inspired from our finding. And so is that real? Is there a real threat in those viruses, which again can be alive for 40,000 years, which in fact makes a continuity between the infectiology of Neanderthal and us, and also between the mammoth. Mammoth disappear. Nobody exactly know why mammoth disappear. There is a lot of different theories, but one could be infectious disease. The rhino also disappeared. So all those things are still mysterious. So can we confirm that result? Yes, we continue to use different sample sites, we use different sample types, and we use different investigation methods. Okay. Thanks to some of our collaborators in St. Petersburg, we got access to the Yana rhino horn site, which is on the Yana River mouth, okay, which is probably the first example that in fact human coexisted with mammoth and probably killed them for food, actually. And so this is a place when every winter there is thousands of pieces of mammoth that definitely go out of the ground, and so it's full of material that you can get frozen and that you can actually study. Thanks to Alexei Tikhonov, the head of the Zoological Museum in St. Petersburg, we got access to a piece of mammoth hair or skin, if you want, that was maintained frozen for 40,000 years, okay? And we put that sample in contact with Aconta amoeba, okay? And we got 4 new viruses, or 4 different families, okay? This is a mimivirus type, Pandora virus, which was the first old one. The second pectovirus, and a new type, Asfor-like. Asfor is a disease of swine, which is quite a problem. So none of that has been published. So this is, I say, it's confidential. Please don't tell us. We also work with the Alfred Wegener Institute, which has access to Yakutia, which is a very difficult thing to do. And we have a lot of corals that I got in Potsdam, okay? And we did metagenomic sequencing, which is we take the whole sample, we extract the DNA, and we sequence the whole thing to see what is there. We sequence about 10 to the 10 billion base pair. We have that many segment of 5 kilobase, which is good enough to make a call, to make a vote of what you see here are the 5 genes of bacteria, viruses, or something else, okay? So it's a lot of information here. And from here you can see that you get mostly bacteria, a lot of totally different bacteria, very few eukaryotes, which is a mystery to me because normally permafrost is made of the tundra, of the shrubs, of a lot of things, but apparently this is decomposed very rapidly by fungi and bacteria. A very interesting thing, which is also extremely confidential, we found that there is also a lot of beta-lactamase genes. Okay, beta-lactamase are the gene that bacteria use to destroy penicillin and all the antibiotics, and those are the main threat of bacterial resistance to antibiotics. Okay, so apparently this is a new threat. You know, this DNA, okay, those genes might actually be transmitted to modern bacteria as a new way of resisting antibiotics. Okay, so this is a work in progress here, but we have more of those than regular genes for some reason that we don't really understand at the moment. Now, viruses. Well, viruses, apparently, most of the viruses we got are the ones we knew about. Most of them are the giant viruses that infect protozoan, amoeba. But there is a lot of other viruses in lower quantity. Again, the numbers are very big. We have about 1,000 million base pair of viral DNA there, okay? And again, the number of elements which are larger than 5 kilobases is quite big, or more than 2,000. So we can make statistics out of it, okay? But we don't see much of pox virus, for example. But again, was there people there? Yes. The interesting thing, the very last mission we have did is around Cherokee, the Duvernay. As you can see, for some reason, people pass through those areas. So there have been human populations, or Neanderthalian populations, or Denisovan populations in this very inhospitable area. During the last glacial maximum. Okay, so if there have been human disease, we could find things there. So just because for you it's certainly quite abstract, it was for me before I went last August. Actually, I went on the site to see exactly how it was done. Okay, so this is Cherokee right here. The Colima River goes this way. This is the delta of the Colima River. You have meanders. So the Colima River, on one side, it's a very flat beach, very fine sand. And on the other side, which is the exposure, this is a small forest, a lot of shrubs. It's very difficult to walk in there because of the ice melting. You don't know where you put your feet. It's very easy to break a leg, so it's not as easy as it looks like. So this is on the cliff side, okay? You have those ice wedges, which are the proof that what is here has been frozen for a very long time. Okay, this is the boat we use, and just to show you that this is really a place where remains of mammoth abound. After 10 minutes, my PhD student, found this mammoth tusk he literally stumbled on it because every summer those things are released from the permafrost. So I'm going to show you a small movie. So again, this is the type of vegetation. So when you go there, you think it's going to be very inhospitable. Well, in summer, it was like about 30 degrees, okay? And my student, not me, they took a bath in the Colima River. Okay? It was about 20 degrees, 20 on the surface, very quickly, much lower than that. Okay? And so this is the other side. So we are going to go to, I hope it's going to work. I want to show you exactly how it is, okay? Yes, great. So this is permafrost. This is like concrete. Okay. Yakutsk, a whole city of 3,000 people, is actually built on this. Okay, so this is, but once it's stored, this is what you have here. So this is real concrete. Okay, sampling, how it is done. Sampling is done with a special tool. This is Stas Malavin from Pushino here. Okay, this is just a special tool we have. Okay, and then we take a sterilized knife, okay, and we cut dices of this at the center of the sample, right here. Okay, we put that. So why? Well, suddenly there's no contamination because before you can do that, you have to remove the active layer. Okay, this is this right here, 20 to 30 centimeters tall. This is concrete, and this is very soft. Okay, so you have first to expose the top. So thawing, yes, thawings. Right here, this is the surface of the permafrost, okay? This is very slow, but what I want to show is that, the whole surface is thawing. That is, the young permafrost, and the old one is thawing at the same time, okay? Everything is thawing. And then it goes into the Colima River. So diffusion is not a problem, okay? It goes in the Colima River right there. Okay. So this is what's going on, okay? I think it's nice to be able to see it, and to be confident in the results. Okay, thank you. So the results again from last August. Okay, 3 new viruses from the Lena River. This is the one that is in Yakutsk. It was not frozen. This is like a test for us. Yes, we find 3 different types of viruses infecting amoebas. Okay, 1 new virus from thawed material, okay, at the Cherokee Donovan Yard, okay, and 4 new viruses from layers of permafrost that are from Pleistocene. Okay, so this is the confirmation that yes, we can actually relatively easily rescue live viruses from permafrost. Those are large DNA viruses. So what is the exact risk? Okay, well, risk is not only danger, hazard, it's exposure. There is a lot of sharks, but if you don't swim, there is no risk for you, okay? So a risk is, first, exposure. Well, exposure will grow, that's for sure, because there are more and more people that are going to work in the Arctic, in those places I've seen in one of my first slides, okay? What about the hazard? Well, it depends on the presence, viability, and resilience of known and unknown viruses, because we don't know who killed Neanderthal either, okay? Because we are always thinking about, old, present viruses, a virus that we believe we could recognize. Well, I discovered in the last 15 years 5 new families of viruses that were not even known to exist. Okay, so I don't think that virology is extant. Okay, there are a lot of new viruses that we can discover that might be dangerous to human, animal, or plants. Okay, so that we don't know. So first, on the precaution level, I think that every large-scale industrial project should include a good medical facility. Okay. And if somebody is getting sick with symptoms looking like smallpox, don't put him in the plane to Moscow. Maybe keeping him to see what's going on, okay? Having a medical doctor that will be able to recognize all disease, because, people might not be, very familiar with smallpox anymore, okay? And actually, real research should be done on real pathogens, because, of course, we work on DNA viruses infecting amoeba because we're not crazy enough, and we're not equipped for working on trying to rescue smallpox, herpes viruses, RSV viruses, or any of those viruses. Okay, this will need the BSL-3 facility. So we use those viruses as a report of viability. But if those DNA viruses are able to survive, I don't see why other viruses, which are, by the way, also cytoplasmic viruses, will not be able to survive that long. Okay, which again make us in continuity with Neanderthal and other animals. The only problem with that, first, we don't have the equipment for that, and not much people have that. And the last thing is that I will not tell you how we do, but it is virtually impossible to legally export infectious sample from Russia, and even more from Yakutia. So this is really something that has to be put in the context of international relationships and things like that, and even internal relationship between Yakutia and Russia, because even Russian scientists have difficulty to get things from Yakutia. Thank you very much.